Okay. Let me actually move this. I don't like its location. Let's move that there and that there. Oh, better. Okay. Oh, now that's off. We'll call that good. Okay. Um, Photosynthesis part 1. This should be lecture 4 in the unit. Let's jump right in. So, photosynthesis will be broken up into two main phases. This video will cover the light reactions, where we're going to split water, release oxygen, make a little bit of ATP, and then also get some high energy electrons. They're back from respiration. Same idea. And then the next video will be on the Calvin cycle, where the actual sugar gets formed and we do the carbon fixation step. So, jumping into the light reactions. First thing I'm going to talk about are these things called photosystems. They'll play a role in the light reactions, as we'll see in just a second. And what they are is just protein complexes, so a bunch of proteins together. You can think of it really as one protein. They're in the thylakoid membrane, and their job is to take light energy from a photon and convert it into chemical energy that we can use in chemical reactions later. So the way that it does this is it has some special pigment molecules in it that are not chlorophyll themselves, but they work with chlorophyll. And these pigment molecules essentially capture some of the energy from a light ray in the form of a photon, just a little bundle packet of light. They capture that energy and they're able to kind of pass it around between them and ultimately to a chlorophyll molecule. The chlorophyll molecule then has the job of bringing the electrons to a reaction center where they will actually leave the photosystem. So this is the first thing really that happens with photosynthesis, is this light capturing process. But it's really just these pigments the electrons get hit by a photon, they get excited, they get energy, and then they get passed along until they get to the reaction center, where they'll be passed to the next step forward. But when you see photosystems, just think, okay, this is the thing that captures the light and turns it into chemical energy. So, an overview, and we'll zoom in to each of these steps in a second. So, the main purpose of the light reactions is to take light energy from the sun and use it to make ATP and NADPH, which is very much related to NAD and FAD from respiration. It carries electrons. It's just a photosynthesis specific version. And so the whole purpose of this is just to make ATP, put some high energy electrons on my NADPH, and then move them out to the next step. So we are first going to talk about photosystem 2 here on the left. So just to kind of reiterate what happens, photon, which is a bundle of light, it's just like a unit of light energy, comes in and excites some of the electrons in those special pigment molecules. The energy gets passed around and ultimately ends up on that P680. It's a special chlorophyll molecule that is receptive to light that is 680 nanometers in wavelength. And in this process, it will also end up reacting with a water molecule where some oxygen is released, as well as some protons that we'll use in just a second. But after that reaction happens, we pass off that electron to the reaction center there, and it moves forward into the electron transport chain. So, electron transport chain is back. It fulfills very much the same role that it does in respiration, where we're using these high energy electrons to pump hydrogen ions against their concentration gradients, so that we can use them to make a little bit of ATP here in just a second. So all that's happening in this step is the electrons are passing through the chain and being used to pump those hydrogen ions. They're just being used as a source of energy. And then they get passed into photosystem 1, where another photon has come in, excites some more electrons that eventually get passed off to P700, which is just a slightly different form of a chlorophyll molecule. But the electron is ultimately headed towards an NADP. It's the special protein. You don't need to know the name. It's called NADP plus reductase. It reduces NADP plus. It essentially, we're giving those electrons to NADP. So the final destination of these electrons that I excited with the photons is that NADP molecule. But all we've done here is we've taken some light energy, used it to excite some electrons, give them some energy, use some of that to pump some hydrogen ions. The rest gets passed off to NADPH, where it is going to head off into the next step. What happens after this is a chemiosmosis step. It's really kind of happening at the same time as the step we just went over with NADP plus reductase. But those hydrogen ions that we pumped using that electron transport chain, just like in respiration, we are going to use them to run through ATP synthase to make an ATP molecule. So this whole process required 2 photons, and what we got out of it was an ATP and an NADPH with some high energy electrons that are going to head off to the Calvin cycle to do some more work. But I wanted to take a second and talk a bit more about the similarities with respiration here, because in a ton of ways, photosynthesis is just the reverse process of cellular respiration. There's a lot of similarities. So this diagram really shows that main one that we just kind of covered, where both of them feature an electron transport chain that uses high energy electrons to pump hydrogen ions against their concentration gradient. If I say pump, it has to be against the concentration gradient, but we pump the hydrogen ions that are then allowed to diffuse back through ATP synthase down their concentration gradients, and we harvest that rushing through ATP synthase like water rushing through a dam to turn a turbine and make some energy in the form of ATP. So in both cases, we are pumping hydrogen ions into a separate compartment, a separate space, and letting them run through. So both of them operate on these hydrogen ion gradients and have these membrane structures so that they can compartmentalize and create those hydrogen ion gradients. Another analogy with these is the mill analogy. You can think of it as kind of like 2 different steps here, where first step, photon comes in. It's like a hammer smacking that, I don't know, seesaw, teeter-totter, whatever it's called. Launches the electron up, gives it some energy, and then we slowly roll the electron down a ramp so that we can turn a wheel that will make some ATP. That's the electron transport chain and chemiosmosis step. The electron continues to fall down a little bit until it gets excited by another photon, a different photon, where it gets re-energized and given to NADPH, which is a bucket where it's going to be carried off to another area where that water could be used to do more work. So in a lot of ways, it is just like pumping water up a hill to let it run down the hill and harvest that energy. Okay, that is going to be it for this video. This next one, we are going to go through the Calvin cycle, which is the second half of photosynthesis. Shorter one, but a lot to digest in it. We're going to work on it in class some more too, so don't worry if you're still a little bit lost. And I'll see you later. Ciao.